Next up, I'm glad to introduce Dr. Christopher Armstrong. Dr. Armstrong is the director of the newest OMF Collaboration Center in Melbourne, which was established in partnership with Emerge Australia in 2020. Chris first started his ME/CFS research at the University of Melbourne through his PhD project that applied metabolomics to study ME/CFS. Now back in Melbourne, after spending some years as a visiting scholar at Stanford, Chris is guiding OMF's exciting new collaboration in Australia. His initial research is focused on an individualized approach to understanding ME/CFS. Please join me in welcoming Dr. Armstrong from the Melbourne collaboration. Chris, thanks for giving us an update. How has the transition from the US to Australia been?
Thank you very much for having me and allowing me to present an update like this. And thank you for OMF for holding these interviews of the CRCs. I think it's important, obviously, to communicate to the community as much as possible. And I'm hoping this is the first of many to come. So just starting with the transition back from the US to Australia, I think that was a lengthy process at the end of last year, but we managed to get back into Australia in December last year and started up with the University of Melbourne in January of this year. Obviously, I'm the director now of the Melbourne ME/CFS Collaboration. And in that role, we've really started to try and get as much collaboration happening within Victoria at the moment as possible, and obviously expanding through collaborations worldwide as well. At the moment, we've gone to first point of call has been to actually bring on new staff. So it's not just myself. So we have a PhD student and a postdoc who have joined the group here at the University of Melbourne. We've been trying to set up a lot of these projects that we have. We're starting from scratch, I guess, for a number of these. That has included a lot of putting together ethics applications, getting approval for these studies, but also going through the protocols and setting them all up and optimizing, I guess, those protocols for the projects that we'll be doing. We have started a little bit of a pilot project in collaboration with Monash in regards to looking at Monash University in regards to looking at a diagnostic, but more of that information will come, not in this update, but the next one as we get more results. We also have a couple of longitudinal studies that we're starting up. So one with adults, but also with pediatrics at the Royal Children's Hospital, which is just down the road from my institute, the University of Melbourne. And so at the moment, we're very optimistic for a big year ahead. We have no restrictions here in Australia. Everything's back to 100% normal as far as I can tell. From that, we can probably expect to have a full year of research unobstructed, I guess. And so that's kind of the big benefit for being here at the moment. But also, I think that hopefully we can get more results out as soon as possible. I think that would be obviously music to a lot of patients' ears.
You mentioned that you're setting up a number of longitudinal studies. Can you explain what they are and why you are taking this approach?
Yes, we do have a few longitudinal studies, I should say. So longitudinal studies are effectively repeated measure studies. So instead of having a case versus control population, which is what we might do more commonly in ME/CFS, and a lot of studies have worked on that in the past, looking at a cohort of ME/CFS patients and a control cohort, and looking at the differences between them. So the difference for a longitudinal is that we're trying to look for more of the repeated or some of the dynamics of the disease within the individual. And so you could potentially do that, still looking at case control, looking at some of the changes within an ME/CFS population versus a control population or another disease population. That's also a way of doing a comparison. But for what we're doing a significant number of repeated measures in the individual, and we're hoping to use the individual as their own control. And what we mean by that is getting a number of samples along a period of time to get a kind of a baseline, and then also get samples at the most severe points of their disease to have a comparison. So we're trying to compare severe exacerbated periods where they have extreme symptoms or worsened symptoms versus periods when they have less worse symptoms or relatively good days, as we call them. And so I guess that's a way of kind of understanding what might be going on within the individual to see if we can find biological changes that kind of represent what's happening when they get worse and what's happening when they get better. So the value of that, we think, is because obviously some of the major ME/CFS symptoms like post-exertional malaise are kind of dynamic symptoms. They're not the sort of thing that you can just say there's one point where you have post-exertional malaise. It's the exertion event getting significantly worse, and then a protracted recovery, I guess, from that. And so I think this is kind of the value of doing this type of research for ME/CFS, but also because this disease is kind of unknown, and it may be heterogeneous. Yeah, so we consider it to be potentially a heterogeneous disease where you may have multiple different disease pathways underlying different patients with ME/CFS. That's a possibility. We still think, obviously, looking for a central component to the disease, but because the disease itself is outlined by symptoms and not with some defined pathology, I would say, it's still kind of not 100% known whether that all the people who are kind of diagnosed through the symptoms with ME/CFS do have the exact same underlying biology. And so that's why we kind of think of looking at longitudinal studies for ME/CFS patients, because we can kind of start defining individuals and then start grouping them, I guess. With ones who look very similar to each other, we can kind of group together and then more deeply characterize that group and compare to other ones as well to see if we can find any subsets of the disease.
It sounds like you consider this to be a heterogeneous disease. Do you not think there could be one cause for the disease?
Yeah, I guess it's not that we consider it to be a heterogeneous disease. It's actually that we don't know whether there is a single cause or there might be multiple. I guess by doing this approach, we're considering that there may be either way. And so we can kind of look for whether it is heterogeneous or it is one component to it. Personally, I think we've taken the approach that we do think there is a central component to this disease, but multiple pathways that lead to that central component. Because the component is so central, I think that means that there is probably more likelihood for multiple pathways to it. And so that's kind of the approach that we take. So I guess we would say we think it's mostly some component, obviously, underlying the common symptoms is probably consistent between patients, but then there's also going to be some variation. So I think that's probably the best way that we can kind of approach it at the moment, since there is no knowns at this point in time. However, of course, we keep an open mind, and as I say, that's the reason why we're doing what we're doing.
Can you describe the nitrogen hypothesis in simple terms?
Yes, the nitrogen hypothesis. So that's kind of what we do think is the central energy metabolism disturbance, I guess, that is common across ME/CFS patients. That's at least our hypothesis. We don't know that, but at this point in time, that's kind of the theory that we're working from. I guess to describe it in simple terms, maybe I'll start with a scientific explanation and then go for an analogy that might help to explain it. So trying to keep it as simple as possible. For energy production in the body, usually required to, I guess, burn carbon to create carbon dioxide. So the carbons will then effectively jump onto the oxygen that you breathe in. You breathe out carbon dioxide. That carbon being split off from metabolites creates these protons, or loads these protons in the mitochondria, and then you're able to utilize those protons in the mitochondria to pump through and create ATP, which is the energy molecule that you use for all types of reactions within your body. And so carbon is central, I guess, for metabolism, for energy, for oxidative metabolism, especially, for all metabolism, actually, for energy production. But so what makes carbon interesting is that, carbohydrates and fats and amino acids all have carbon in them. And what makes them different, I guess, is that for amino acids, what makes them more unique is that they have the nitrogen molecule, whereas carbohydrates and fats do not. And so they're also a very useful form of fuel, I guess, for producing ATP. But in that process, the body has to remove that nitrogen from those metabolites from amino acids. And so when you're more heavily using amino acids for ATP production, you're producing more of these nitrogen byproducts. And some of them can be quite toxic in certain types of accumulation. And your body has, or your cells have ways to remove that nitrogen from the mitochondria. But if you're more rapidly using it, you may not be able to, I guess, remove it as fast as you would like. And that's really the basis for the theory of this, which is that more amino acids are being utilized for energy production, or effectively more nitrogen is accumulating, or nitrogen byproducts are accumulating in ME/CFS patient cells when it comes to energy production systems. And so they are having a kind of a toxic effect. And so I guess you would say that the basic understanding is that through this mechanism, the production or creation of energy is actually creating a problem in ME/CFS patients. One of the most initial problems of nitrogen is the accumulation of what's known as ammonia, which can actually pretty much stall energy production pretty rapidly. And so the more that you produce energy, the more you're creating this ammonia if you're using amino acids, and the more that this ammonia accumulates, the more likely it is to pretty much slow down the energy metabolism and pretty rapidly as well. And so we think this could be a kind of a basis for a number of symptoms with ME/CFS. Obviously, ammonia and nitrogen byproducts are also being a long literature history of being attributed to issues of cognitive dysfunction, brain, things that would indicate brain fog, but also you have encephalopathies and all that sort of stuff. This is kind of different, and this is not the sort of thing you would expect to find as high ammonia in the blood for our hypothesis. This is what we're actually indicating is that you have, I guess, an accumulation of nitrogen within the cells themselves, okay? And so I guess if you were to use an analogy for that, if you have the sewage being indicative of the blood, and then you have a problem, I guess, at the sewage treatment plant, this is be more like what we think of like when you have an issue with your liver. Then you start to get, I guess, an accumulation of toxins throughout the sewers because the treatment plant isn't removing all those toxins. And so that would be like ammonia building up in blood. And that's when you have something wrong with your liver. You get elevation of ammonia. They can test that at the doctors, and usually that's to do with something like cirrhosis or some type of liver problem. I guess in this example, if you're having issues at the point of the houses, like in the bathrooms, or there's all the toilets suddenly start breaking, and everyone, like the sewage component of that being built up in the houses, you're not going to see it within the sewage plant. You're going to see it at the homes, and that's kind of like the cells, I guess, in this analogy that I'm talking about here. Some interesting aspects of this is more common in females. Females are more rapidly utilized amino acids than fats. That has a lot to do with actually female composition being higher in fats and lower in protein compared to males who are higher in protein and lower in fats. And a lot of that is to do with sex hormone differences. And so females are more rapidly utilizing amino acids already. And there are a number of things that can contribute to this, not just the increased use of amino acids, but also the decreased ability to remove nitrogen byproducts from the mitochondria or anything like that. And so that's kind of like the idea behind this hypothesis, I guess, more scientifically. And you may wonder like how we think that kind of comes about. We think it could very well be that there are genetic issues within nitrogen removal, or at least increased amino acid consumption. Pathways that lead to that may be certain dysfunctions in the use of sugars or fats, but also could be dysfunctions in the ability to remove these nitrogen byproducts as well that get produced. And so obviously issues in fats and sugars would lead you to use more amino acids. And so we think that is kind of a way of pushing towards the use of amino acids as well for energy production. And I guess a kind of an analogy I like to use, and I think it works pretty well for describing what we're talking about. You could have had a house, I guess, and you have a petrol generator as a backup for energy production in your home. Now you have on-grid power, And I guess you're going through your entire life. You have on-grid power, and there might be periods of time where you get shutouts of that energy, and you're going for the backup generator. Now, I should add that under stress, you increase your production, or you increase the usage of fats and amino acids. So usually the body likes to use carbohydrates for energy production, but you increase the amount of fats and amino acids for energy production under a stressful condition. And so we like to think of this as kind of like a backup generator during stressful periods. You start to use your petrol generator effectively, different types of fuels to create energy, I guess, within your household. And then maybe it's working fine for little short periods of times. You have a cold, you have other stressful events, and the backup generator can do the job. But then suddenly, you have a blackout for a long period of time, maybe a month. You start using the petrol generator, and there's something wrong with it. There's always been something wrong with it, but you never knew because you never used it long enough to realize that there was an issue. And so what I'm talking about is whether you have genetic anomalies within that backup generator pathway, within the amino acid or fat metabolism pathway, and within the nitrogen removal pathway. So you're using this nitrogen, I guess, this petrol generator for a period of time. Perhaps it's spreading fumes into the household from the petrol generator, and you're starting to get quite unwell because of that. I guess that's the analogy, I guess, that we're running with in terms of a potential explanation for how this could crop up for people at a later stage in their life, not just something that they're born with. And so that's kind of, I guess, the best or simplest description of this. We have written up a lot more detail on this hypothesis and into a paper, which we will be trying to publish in the very near future. I'm sure people will be very interested to hear more about that.
That's an interesting hypothesis. How do you plan to test it?
Our plan for testing this hypothesis is actually going to come from trying to measure these nitrogen byproducts. So both in cells, so taking cells from MECFS patients and tracking the cellular metabolism, and specifically nitrogen-labeled nitrogen substrates. So we can feed the cells substrates that are specifically labeled for the nitrogen and look where the nitrogen goes through MECFS cells versus healthy cells. That's one thing that we'll be doing to test it. Also, through these longitudinal studies, we'll be checking through the urine. So most of the time you remove nitrogen byproducts through your urine. And so if we can track over time, it might be too subtle to track one sample, but we plan on tracking 24-hour urine, maybe over several days, and just determining if we can find a link between increased nitrogen output and energy usage that is more exacerbated in MECFS patients than controls. And so those are the two ways. One is, I guess, an in vitro measure and not incorporating environment. So in vitro is the cellular, growing the cells and looking for cellular metabolism. That won't tell us much about whether there are environmental factors. More, it's just going to tell us if there are genetic factors leading to potential increase of this nitrogen byproducts in the cells. The in vivo measures will be the longitudinal, where we're able to get a total output from the individuals to see if they're getting an increased output of these nitrogen byproducts, potentially from urine over time. So yeah, that'll be the main ways that we're going to be testing that.
We'll all be interested to see how that turns out. Thank you for taking the time to update the community, and we look forward to hearing from you soon.
No problem. Thank you very much for having me. We'll check in soon.